Hello, and a very warm welcome to today's Nucleic Acid Insights webinar, titled Advancing Nucleic Acid Therapeutics, Scalable Strategies for Plasmid DNA, mRNA, and LNP Purification Supported by Rapid At-Line Analytics. I'm Charlotte Barker, an editor at BioInsights, and joining me today are Rok Sakernik and Tristan Kovacic, who will outline how targeted process choices at each stage of nucleic acid therapeutics can significantly improve yield, reduce impurity burden, and simplify downstream development. After the presentation, we'll have a live Q&A session, so do pose your question to our speakers using the Ask a Question box at the bottom of the screen, and we'll try to get to those during the session. And I'll just briefly introduce our presenters. Rok is Head of Process Development for mRNA and pDNA at Sartorius BIA Separations. Rok's team is focusing on developing high-yielding, cost-effective purification approaches for pDNA and mRNA production. Tristan is a project manager for process analytics development at Sartorius BIA Separations. He's responsible for the development of applications of non-viral drug delivery systems, including LNPs. His expertise spans analytical development, purification, and manufacturing, with experience in biomolecule processing using monolithic columns. So without further ado, I'm going to hand over to Rok to kick off today's presentation. Over to you, Rok.
Thank you, Charlotte, and thanks to everybody who's dialing in today for this webinar. We have quite a lot of ground to cover because we would like to show you everything we know about purification and scalable purification of both plasmid DNA, mRNA, and LNPs. So without further ado, I'd like to jump in. First of all, where we are streaming this from. We're located in a facility in West Slovenia, not far from Trieste, where there's a red dot drawn here. And when we look outside of our laboratories and offices, this is what we see. We see vineyards. If we drive 30 kilometers down the road, we see one of the most magnificent caves, Postojna Cave, which is also part of inspiration for the monolith technology that we use in our daily work for the purification of large biomolecules. So if you're ever in the region, please do pay us a visit. Today we focus on non-viral delivery systems, so plasmid DNA, mRNA, LNPs, but we also work on gene therapy vectors like AAV, lentivirus, and so forth. We would really like to take you through the entire workflow of what it takes to come to a highly purified LNP to deliver a nucleic acid payload, but we have to unwind the story quite a long way back, and we have to load the LNP with mRNA, but before we get to mRNA, we have to actually make highly pure plasmids. And so one of the basic tenets of this webinar is that production of mRNA/LNP starts with high quality plasmid DNA. And I will walk you through not quite all the steps in this workflow, but selected steps which I thought were interesting for the purpose of today's presentation. Now, why should we worry about purity of plasmids? There's increasing body of literature emerging, particularly in the last 1 to 2 years, which suggests that poorly purified DNA template, or less than ideally purified DNA template, can both give rise to impurities in the downstream process, including in vitro transcription of the DNA. There's one paper referenced here that shows how impurities in the plasmid DNA prep were leading to double-stranded RNA formation. Another interesting paper went a step further and actually started to identify that nicks in DNA templates, so-called open circular DNA impurities, of which I'll talk about in a minute, also can give rise to aberrant mRNA production, which can then carry through if we don't apply the right tools down the road. So high plasmid purity, and here we really are now addressing the high finesse of plasmid purity, so minimizing even the nicking of DNA is becoming increasingly important. And so this is an important part of our job when we develop plasmid DNA processes, is to provide with methods as well as the tools to get to this high quality of plasmid. What are the challenges that we have to tackle along the way? First is the challenges themselves in fermentation, how to get to high yield. I'm not going to address this. We have other excellent teams working on this, but already at the point of lysing DNA out of cells, you can face significant challenges, particularly in scale-up. Clarification can be a challenge. If the lysate is of poor quality, we have to process relatively large volumes, and then we have to efficiently, meaning with high recovery, get out only the pure stuff. So these are all the challenges that we tackle daily, and we try to provide elegant, scalable, and GMP-compliant solution as part of our daily work. And so I'll show now a couple of tools that we either developed or used to get to our desired goal. So highly pure, and by highly pure, I mean 98+% pure supercoiled DNA without endotoxin, without host cell RNA, and so forth. Some of the tools, so both equipment and consumables that we use and that you can use along the way, are widely available through our parent company, Sartorius. I here show a couple of examples, both for lysis, for clarification, for diafiltration, capture, polishing, and so forth. All the tools are at your disposal. It's just about getting them connected in the right way and used under the right conditions. But the subtitle of today's webinar is also Fast At-Line Analytics. And here I want to showcase one machine in particular, one of our favorite machines, and this is so-called PATFIX system. It's a biochromatography system that we designed as process developers, I can say, to fit our needs for rapid at-line analytics. And our philosophy is very much to have this system embedded in process development workflows such that as a process developer, we get information on the quality and the quantity of our target molecule ASAP. And by ASAP, we mean minutes. And I'll be showing examples throughout, and then Tristan will pick up and take this forward also in the field of LNPs, how we can really benefit from having a PATFIX system embedded in our process development and quality workflows to give us the information we need when we need it, and not a week later. Before I get to that, let's just ask a very simple, basic question. I should like to add one more thing, is that everything that I'll show here is the result of wet lab work performed in real laboratories with real people who are my excellent colleagues within our labs. And so all this process knowledge is available also for partnering. And we'll be delighted if, as a basis of this webinar, you reach out and we can run a project together. It's not just about the tools, it's also about the process knowledge. So don't be shy, feel free to reach out and we can talk about process optimization. And now to the question, what is this molecule we're even talking about, plasmid DNA, that is so important for production of mRNA, also for production of gene therapy vectors like AAVs. Plasmid DNA is really the foundation of modern biotechnology and modern biopharmaceuticals. Plasmid DNA is a molecule that we find in bacterial cells, typically circular double-stranded supercoiled DNA of around 5 to 10 kilobase pairs, but we quite often also work with 15 kilobase pairs, even up to 25 we've tackled successfully as a downstream process. You can see already that it's well hidden within a bacterial cell. So before we can purify it, we have to actually open up the bacterial cell. That's challenge number one. And second challenge is down in the right-hand side, you see 3 isoforms of plasmid DNA. One's called supercoiled. That's the target. That's the one that's actually most efficient at transfection. If one base pair breaks, the supercoiling starts to relax and we get so-called open circular or nicked DNA isoform. That is less desirable, or it's, I mentioned earlier, potentially undesirable. If the nick happens in the coding region, then we can get shorter mRNAs, we get aberrant mRNA formation from it. And then the next, if the other strand is nicked again, then we can get linear isoform. Now this is not necessarily an undesirable isoform. In fact, for mRNA production, we quite often linearize our plasmid DNA. It would be great if plasmid DNA was all that was found within bacterial cell, but unfortunately, plasmid DNA is on average only about 3% in a given fermented biomass. Everything else is either proteins or host cell RNA, endotoxin, the toxic component. And so we have to isolate at best the 3% of material, and 97% of biomass is impurities. How do we release it? There are various ways, but since mid-80s, one method in particular has been the industrial workhorse, and that's so-called alkaline lysis, simply meaning that we have to incubate bacterial cells with strong base, sodium hydroxide mixed with SDS, and that leads to breaks in the cell wall as the first step. That opens up the gateway, if you like, for plasmids to be released. But soon after, the same sodium hydroxide also starts to denature DNA. It can start to shear up genomic DNA and start to produce relatively unpleasant impurities that we then have to tackle in the downstream process. Traditionally, alkaline lysis has been performed in so-called batch mode. This simply means that cells, resuspended cells, were mixed with lysis buffer, with sodium hydroxide. Then neutralization buffer was added to it, and then precipitating agent was added. Everything was stirred all the time. Now this is a procedure that we use in lab scale experiments. This is how you would first do your DNA isolations as a student, but it quickly becomes very problematic when you start to scale this up. Imagine pouring 100 liters of base onto 100 liters of cells, and you can start to imagine there are some issues with control of this process. So the idea of so-called inline lysis or continuous lysis has been quite important in the plasmid industry, but with a distinct lack of tools available to perform inline lysis. And so over the last few years, we've really stepped into this field to enable the plasmid DNA producers to use a reliable device, machine, to support inline lysis mode. Why is this important? First reason would be a seemingly trivial one. If you perform batch lysis, neutralize and flocculate your material, you get the product of the quality on the left-hand side. You quickly see it's a mess, and you can imagine how it is to filter this mess. You clog up all your filters. Whereas if you perform inline lysis, continuous lysis with this, shall we say, the modern way, then the filtrability of the sample is significantly improved, partly also due to airflow that the machines enable. And you can see now the flocculation is lifted nicely to the top, and you can filter only the bottom part of the sample. But it's even much more important than that. Inline lysis not only preserves the recovery or enhances the recovery of the plasmid compared to batch lysis, it also significantly reduces the release of some of the, let's call them, nasty host cell impurities. We've shown time and again that endotoxin burden in the lysate performed with inline lysis is lower, significantly lower, as is the contamination with genomic DNA. Up to 5 times less genomic DNA contamination is found in samples that are lysed with continuous lysis. And this has now been verified not just in our laboratories, but in laboratories that are running our machines throughout the world. So what is this fantastic machine that I'm describing? We called it alkalizator, the strong name because it performs one of the critical functions in the plasmid. DNA downstream process. It's currently available in 2 sizes: 1 liter lysis reactor, 5 liter lysis reactor, and this is sufficient to service really a up to 10 to 50 liter bioreactor volume, so fit for the majority of plasmid DNA needs that we currently see in the market. So this has been an answer to a lot of headache that we have seen in the field of recent. Then I've shown how the filtrability of the samples is improved if we perform inline lysis with alkalizator. So I'm now going to skip through the clarification question. We have excellent filters available, and we can talk about that some other time. But I would like to highlight the next important step in DNA isolation, which is DNA capture, for which we have traditionally used a weak anion exchanging column, a so-called simultus DEAE monolith. You can see an image of monolith chromatographic columns on the top right-hand side. The DEAE version is excellent for capture of plasmid DNA, also for removal of whole cell RNA, and I'll show an image in a minute. It significantly reduces proteins and endotoxins with a high binding capacity. 7 grams per liter is quite standard, and it comes in 2 channel size flavors, which I'm not going to go into more detail, but depending on the size of your plasmid, we can suggest with 1 pore size or another. And so this sort of chromatographic column is used really to purify any plasmid DNA from 3,000 base pairs up to 25,000 base pairs. We have purified with this. And how do we know if our purification works well? Well, I bring back that first or second favorite machine that we use, so-called PATFIX analytical system, which we run in parallel with our purification. On the left-hand side, we see a preparative chromatogram, samples coming off a DEAE preparative column. On the right-hand side, we look into individual fractions with PATFIX analytics, and we see that the so-called RNA wash that we perform really washes off all the residual RNA. We see that the elution contains more or less exclusively plasmid DNA, and we see, for example, also what is the ratio of supercoiled to open circular in our elution. This column doesn't fully remove the open circular. In order to do that, to get really highly purified supercoiled plasmid DNA, we use hydrophobic interaction chromatography in a flavor that we call SHIC, which stands for selective hydrophobic interaction chromatography. And there we developed a method which is highly selective for supercoiled DNA, such that our HIC column only binds supercoiled. Everything else flows through the column, and so our elution contains more or less, I will call it nearly 100% supercoiled, but nothing is ever 100%, so let's call it 98%. This is what it would look like analytically. You can see if the load contains some small amount of open circular, the open circular ran right through the column. It didn't bind, whereas our supercoiled did bind. And so that was the only isoform present in elution. And if for whatever reason you didn't trust chromatography, you can see the agarose gel on the right-hand side, which says the same story. And so if I draw the line, we then have to buffer exchange this material and filter it. I'm not going to go into this. There are technical materials available, but I would like to say that with our process, as I described, our total recovery are quite high. We see consistently between 63, 64% recovery, which is higher than what we see among alternative providers that present their processes. And even if there is a slightly higher recovery that we see, when we look at the purity of the material, we can see that the material quality coming through our process is really high. And so that sometimes certain processes that claim slightly higher recovery actually then show, let's say, higher endotoxin burden in the elution. So we claim we have the best ratio between recovery and plasmid DNA purity that you can find. And so this is really our this is what we take into then the mRNA flow. So we have traveled a long way through this process chart already. We made our supercoiled DNA. Before we can start to produce mRNA, we just have to linearize it, linearize a supercoil. We use a restriction enzyme, depending on the construct, BSPQ1, ND1, whatever it is, whatever your construct is compatible with. And instead of just following linearization with agarose gel, we again bring in our rapid at-line analytical system, PATFIX, and we can start to monitor the linearization of supercoiled DNA into linear with identical chromatographic method. We didn't have to adapt, we didn't have to develop a new analytical method for this purpose. No, we use identical analytics to support us all the way up to here. So we can plot area under curve of our chromatographic peaks shown on the left-hand side. We can monitor the conversion of supercoiled in near real time with some short analytical lag. We then plot area under curve, integrate, and we can really get an excellent view into the kinetics of our linearization. Why is this important? You know what the price of linearization enzymes is. It's high. So every unit of enzyme that we can decrease makes a massive COGS reduction when you scale up. And so to be able to quantitatively assess the relation between concentration of enzyme required versus conversion rate makes a big COGS impact. So analytics, rapid, high quality at-line analytics is very important. And I do need to eventually hand you over to Tristan, but I would still like to cover a few important things about how we now turn this DNA template into mRNA. I don't have time today to talk about IVT reaction, other than it's a complex multi-component, but a cell-free reaction that turns DNA into RNA. And once again, we will come to the power of at-line analytics performed by analytical monolith chromatographic columns, which maybe just to remind you why these are such rapid and high resolution columns, because they are essentially an interconnected network of channels without any dead end pores. So diffusion plays no role in the mass transfer. We see exclusively so-called convective mass transport, which means that our analytics, as well as prep actually, is rapid, high resolution. And this is an example on the right-hand side of a typical analytical run where we increase the flow rate with a monolith. We see we lose no resolution independent of how fast we ran the analytics. And in context of mRNA, which is produced out of building blocks, the NTPs, templated by DNA, we've, I believe, pushed this to the limit where we can now resolve our IVT components, NTPs, mRNA, plasmid DNA, and capping reagent in less than 3 minutes. In less than 3 minutes, I think this is the fastest analytical method available for this work in the world. We can look into the reaction and see how much mRNA we have at any given time, how much residual nucleotides we still have, and how much cap we have. And we can start to plot this as a function of time. So we can start to see how our mRNA is produced over time and how NTPs are depleted. Now, this is an example that's quite a few years old. Over time, we have done many studies of how we can now optimize IVT reaction. Again, I don't have time today to go into all of the details. A lot of this is published in the references I give down below. But an interesting story is the middle one, where we turn Ember 250 bioreactor to continuously produce mRNA. And from 100 milliliter starting volume, we produced over 2 grams of mRNA. And then we have added digital tools into the game. We start to couple PATFIX analytics of IVT reaction with digital tools, particularly MODE, design of experiment software. And we really started to understand what drives the IVT reaction to the point where we could break the world record in IVT yields. So 25 gram per liter is, I believe, the record. But we did the reaction not to make so much mRNA, but rather to prove that, as is described in the paper with the reference below, that we really understand the drivers of IVT yield. Please follow the story or reach out if you'd like to learn more. The message is it's possible to really track what are the important parameters in the IVT reaction. It's possible to push the yields to levels that are previously not been considered possible. And what's even better, it's possible to then purify out this mRNA from this high yield reaction with simple tools like oligo DT. Oligo DT is a workhorse of mRNA purification. It's simple affinity-based method that targets the poly A tail that mRNA needs to have in order to be biologically active. And so we've got, the industry tells us, the best oligo DT chromatographic column on the market because it's fast and it's high resolution. And so you can expect to get these nice sharp elution peaks at high loading flow rates. So you actually save time in the loading phase and you get sharp elution peaks. So you even concentrate down your mRNA in the elution. And all you have to do is apply a little bit of salt in the loading and then take the salt away, which leads to elution of mRNA. An important paper in this development is listed down below if you are interested in more details. And just before I hand over then to the continuation of the LNP story, I should mention also that we are tackling also very seriously this important question of double-stranded RNA, which is the undesirable byproduct of IVT reaction. There's a lot of science currently done on optimizing the IVT reaction so as not to produce double-stranded RNA, but we see that people still would prefer to have a safety net, if you like, a purification tool that would remove double-stranded RNA in case it was produced. And here, let's say the traditional, highly reliable tool is reverse phase chromatography. And we have done a nice study that rebuked some of the myths about reverse phase chromatography, meaning which stated that it's not scalable and that it's low recovery. We've shown that neither of those two things are true. We can scale to 8-liter column volume, no problem, reverse phase purification in step with high recovery to remove double-stranded RNA, but also fragmented RNA and even residual templates. We can actually replace DNA's treatment in mRNA process with reverse phase chromatography. This is also published. I invite you to read the paper. The effects on mRNA activity were quite staggering. We were able to show that really, if you start with wild-type nucleotides in your IVT and produce some double-stranded RNA, let's call it an average IVT, that you could really boost the activity of mRNA if you purified it with this reverse phase. We show this both on mRNA system and on self-amplifying RNA system, which is an even more complex way of making it. And then finally, as let's say the cherry on the cake, I do want to share a most recent story coming out of our labs, and it's an answer to the question, yes, we see that it's important to remove double-stranded RNA, but we don't like the use of acetonitrile. So we really were thinking hard and asked ourselves the question, double-stranded RNA, well, what holds it together? And we came to the conclusion that what holds double-stranded RNA together is hydrogen bonding between the base pairs that we all know so well. And so we built now a process on very simple concept that what if we gently disrupt this hydrogen bonding between G's and C's and A's and U's by not by temperature, which would be done, for example, in a PCR reaction, but by mild reduction in pH. So if we protonate certain key hydrogen bonding acceptors, we actually disrupt double-stranded RNA formation. And this is exactly what we were able to show by any double-stranded RNA detection technique, either J2 dot plot or other newer kits. And we were also able to show that by doing this in a relatively short space of time, we do not impact the integrity of RNA. In fact, when we couple this treatment with oligo DT capture, so we disrupt double-stranded RNA by pH modulation, which leads to all of the strands that were previously forming double-stranded RNA, they're now floating in solution, and then we put oligo DT as a selective ligand only for full-length polyadenylated mRNA, and we pull out just the target. But what we see is a couple of things. If our loading material still contains some J2 antibody signal, the elution quite consistently doesn't. It's double-stranded RNA free. And what fragment analysis shows us is that we are really removing anything that is not polyadenylated, but we're also removing very long double-stranded RNA structures, such as 3' extensions. This is work that you'll be able to read about, hopefully, shortly. The final thing that I should like to say about it is that the impact on biological activity of mRNA purified in this way is, again, quite dramatic and exactly like what you would expect to see if you're removing double-stranded RNA. In a cell line that doesn't detect double-stranded RNA, we see no big impact on cellular activity, whereas in an immunocompetent cell line, A549, we see a massive increase in mRNA expression, and we see a massive reduction in interferon release. So with that, I just point out to one final feature of this, which we're also learning about, which is that whether we then use this at very small scale, a couple of micrograms, or at milligram scale, or at 50 milligram scale, we do not compromise on the recovery of this isolation method. We get consistent high RNA yield of the column, 90+%, and we consistently see a negative dot blot signal for RNA that's been purified this way. And we apply this now to self-amplifying RNA, so anything from 1,000 nucleotides up to 10,000 nucleotides, and it works quite consistently. So that's one of the methods that you're due to hear a lot more about in the future. In interest of time, I will say massive thank you to a wonderful team that has put a lot of work into this. I'll stay online for more questions, but I would now really like to give the stage to Tristan, who will now formulate this mRNA, put into LNPs, and monitor the process. So Tristan, over to you.
Thank you. Thank you, Rock, very much for brilliantly starting this story about the drug substance that we use in this novel nucleic acid type therapeutics for other therapeutics, for vaccines, for CAR T therapies, or for other applications. So let me now go into the non-viral delivery of such drug substances. And first, I will spend a few minutes just talking about why we need to deal with our non-viral delivery vector in a way that I am suggesting here. And first of all, it is an inherently very complex product. So we take our drug substance, our mRNA, or our DNA, or our other type of nucleic acid, and we need to encapsulate it into a sort of vehicle. And in recent years, the most common one has been a vehicle called LNP, lipid nanoparticle, on which I'm going to focus on today. But a lipid nanoparticle is basically just an assembly of lipids and of nucleic acids that then forms this nanostructure that is then able to go into the body, protect the nucleic acids from degradation, and transfect into the cells to create the desired either protein production or gene silencing or gene editing or so on. And first of all, as I mentioned here, and Rock already had a big story about, is the purity of the drug substance and its intermediates is key here. So you already want to have your drug substance as pure as possible, and you want to produce it at a sort of in an economical manner, and for then it's to be encapsulated and used further in this drug product. And this is obviously the first challenge already addressed today. But then you're already now introducing your excipients, which are in this case your lipids. In the case of LNPs, your ionizable lipids, cholesterol, phospholipids, and pegylated lipids. And these are again new molecules that need to be controlled and introduced there. Once you form this particle, you then have a relatively unstable assembly, as it is only held together by intramolecular forces. And this then you need to take care about during your manufacturing and during your stability with different approaches to be able to control the stability. And for this, it is very important to use analytical methods, appropriate, comprehensive analytical methods. Again, because of a complex chemical structure, and also because of the sort of diversity that you can use different types of lipids and so on, you are creating a huge heterogeneity across different samples and within the same sample. And these interactions between different molecules in your drug product create then also interesting chemical environments within that drug product, and that can lead to certain impurities, instabilities, and so on. So in terms of what I want to focus on first in this case is the CMCs, and especially the characterization part of this of the lipid nanoparticles. So I'm showcasing here a USP guidelines for mRNA drug products, which is currently drafted in this sort of manner. So we have, for example, ribogreen or fluorescent-based assays for RNA concentration encapsulation efficiency. We have some lipid content analytics, and we have LNP sizing and some purity assays. Now, those are slightly limited type of methods. So we will, as we progress through to a more commercializable product, as we progress through different cycles of optimization and manufacturing development and releasing of such drug products, we will need to expand this analytical tool to truly cover all the bases and to cover all the possible characterizations that we need to properly predict how our drug product is behaving. And I will just mention a few missing key points I believe are missing right now. If we focus on this type of draft guidelines, and one of them is the multiple cargo formulations. So once you have multiple cargos within your drug product, meaning, for example, CRISPR mRNA plus guide RNA, mRNA plus DNA for certain CAR-T applications, or then, for example, multiple RNA product for combination vaccines, you will need to address that heterogeneity, which is then even higher than with just a single nucleic acid. You have some, as I mentioned, there is a different chemical environment within the LNP, so you have actual chemical impurities forming, which are not often looked at such large biomolecules. So that is one of the challenges as well. Then you have certain downfalls of the existing methods for this type of formulations. So where you have different cargos inside that you need to separate, where you have a higher heterogeneity and so on, that you need a method to address that. So that is where we will need to work on new methods. And then we come to a even more complex system, which is now also a very hot topic and in development, are the conjugated LNPs. So LNPs that have targeting ligands on the surface that then work to actively target tissues in the body. Because first of all, we want to be able to achieve the response that we create by introducing the nucleic acids. But second of all, as in therapeutic manner, we want to be able to also target certain organs in the body. And for our active targeting in a sort of relatively straightforward manner, we then can introduce targeting ligands to the surface, but that drastically increases the complexity of the LNP system. And then on the other hand, we also have some endogenous targeting we can exploit in this case. And this leads us also to the last key missing point here, and is then LNP stability in blood or in the body. What happens to the nanoparticles once they enter the bloodstream, once they, let's say, change identity, once they become even more unstable in that type of environment. So, Rock, already described most of this. So our process for end-to-end manufacturing from E. coli to LNP, and I'm going to focus today more on the last part. So LNP analytical workflow and the LNP purification workflow. So in terms of LNP analytics, to cover some of those challenges that I have described before, I want to introduce just briefly some of the methods we developed and that we believe can help you develop your LNP product better and in a more robust way, and gain more insight into what you already have the data for. So the first would be our product called the PodFix LNP Switcher. It's another platform in addition to the DNA and RNA that Rock already described to cover the whole process for non-viral drug delivery. And here we work with LNPs as sort of direct injection of them to our chromatographic system. So we utilize the advantage of the monoliths to be able to perform intact chromatography of LNPs. And we utilize one column to selectively capture the LNPs and the other one to sort of analyze the nucleic acids. And with this, we can achieve determination of encapsulation efficiency. If we have co-encapsulated cargo, I mentioned before, we can separate the cargos between each other. We can determine the size of the particles. We can determine the integrity of nucleic acids. But additionally, we can also detect impurities like nucleic acid lipid adducts. So this is the setup. So we utilize one hydrophobic interaction chromatography column, the OH column, to selectively capture the particles. And then we sort of the free nucleic acids or your free targeting ligands, they don't bind to this column and they will go past and they will bind to the second column, where they will get eluted and analyzed using reverse phase ion pair chromatography. And this type of chromatography, we're able to detect the nucleic acids, we're able to quantify them from a calibration curve, and we're then also able to say something about their quality, because we can do sort of RNA fragmentation study with that type of column. In the second step, we are releasing the particles from the first column. They go past the multi-angle light scattering detector, where we detect their size. And then here, particles are also briefly separated by size. I will showcase some of that later, but the OH column can separate particles by size because larger particles are interacting stronger with this type of column. And then we are doing an inline opening of our particles, so we don't need any detergent or any reagents to be able to open this particle. We utilize the inherent properties of very hydrophobic surfaces like the SDVB that are able to lyse and to open the particles in this manner. So we are directly collapsing the particle structure on the column, and then we are eluting separate components down from the column, so the nucleic acids, the lipids and some of the impurities. So this creates for us a chromatogram like this. So this is again from the PodFix LNP Switcher, the tool that gives you all this data, and you're able to then constantly analyze your LNPs at all steps of your process to be able to properly control what happens, where it happens, why it happens, and so on. So, for example, here we have a combination formulation with RNA and DNA, and it is very nicely separated between RNA and DNA, and then also the adduct impurities, so we can properly determine encapsulation efficiencies, concentrations of both of them separately, and then of the adduct impurities as well. In the multi-angle light scattering, we see a peak that is sort of heterogeneous, and if we plot the radius here with this pink line, we can see the increase in size, and so we can see this size heterogeneity as well. The fronting of the RNA peak can give you fragmentation data of the RNA. That is something you can find in our library on how to do this. My colleagues wrote an article on that, and that is also one of the data you can get as an orthogonal number to the capillary electrophoresis data. To spend just a minute on the RNA lipid adducts, which we think are very important impurities formed within these formulations. This is because of the special chemical environment within the formulations, and we are forming these covalent impurities between nucleotides and lipids, which means that we are decreasing the efficacy of the RNA within the drug product. This has been shown in some articles. There's a few different structures of these impurities available. There's different mechanisms going on, but they are impurities that are then much more hydrophobic than the RNA because of these lipid tails from them, so we can nicely analyze them on the reverse phase chromatography, like the type we are using here with the SDVB monolith. And because we are here using direct opening, so we are not extracting this RNA, we get a more accurate number for these adducts because we can see them directly. We're not losing them anywhere during the process of extraction. Recently, we also wrote a review article in Nature Views Chemistry that deals with such chemical impurities within LNP, so you're welcome to check that out. Now, to move slightly further into a more complex mixture, let's talk briefly about conjugated LNPs and what are the challenges within that field. That field is definitely has a big challenge of CMC because of all the different processes that you need to do to form such a product. So in addition to the sort of unstable LNP, you are now introducing targeting ligands to the surface. And while you're doing that, you want to make sure that you are conjugating everything, that if you want to analyze how much of the protein on the surface has not successfully conjugated, you want to purify it away. Then you want to be able to check in your particles if all of the particles have those targeting moieties on top or if they don't. So and then also you want to say something about the orientation and the stability of such particles and surface ligands, so that you can properly monitor your process of manufacturing such systems. And to do this, we utilize a hydrophobic interaction chromatography. You're able to monitor the conjugation reaction by not binding the free proteins, and this way you can sort of monitor how well this reaction goes. And here you see your intact particles, and you can use the methods to monitor that, and then also you can use it preparatively to remove your non-conjugated surface ligands. Additionally, we developed one ion exchange method that is able to separate between conjugated and non-conjugated particles, or targeted, as I mentioned them here. So targeted particles under these conditions don't bind to the column, while different targeted formulations have different amounts of targeted formulations and untargeted formulations. So this is also a novel method in this case that you can use to monitor your conjugation reaction, or to polish your formulations, which is very important and is first here. We are first here in having a method for intact chromatography like this of these types of formulations. And then to briefly move into the larger scale of the LNP. So I focused until now on the analytical side, but monoliths are highly scalable, and we want, and as is the title of this presentation as well, we have scalable strategies for that. So a lot of the methods we develop can be on an analytical scale, can be scaled up, or the other way around, so that we can use them for purification of LNPs. Here, especially, we're talking about the OH column that I described in the 2D analytical setup. This column, we have now come to a stage where we can sort of robustly use it to capture the particles and release the particles from this column, and for it to be an alternative to TFF or dialysis. But dialysis is used at small scales and TFF is used at large scales. Here you can use the same process for all scales. So you can use the same process for at a lab scale at the screening stage, and then you can use the same purification process when you scale up to a very large scale. And while you're capturing the particles on this OH column, directly after encapsulation, with some inline dilution with the loading buffer, you also get rid of your free nucleic acid, so you enrich the encapsulation efficiency of your ethanol. So then you can sort of elute in your formulation buffer and collect your particles in a concentrated manner with the buffer exchanged and the purified of your free nucleic acids. So this is how, for example, a chromatographic run looks like. Then in interest of time, I'm going to go a little bit fast here. But yeah, you can monitor the purification with UV and multi-angle light scattering, and then collect your particles there in a concentrated manner. So we have tested and compared this data with TFF, for example, and we robustly see that the sort of TFF retentate, when we do that for LNPs, the size distribution of those particles is not as narrow as it is in the case of the OH elution. We believe this is because the monolith is a shearless type of process, so we have very, very low shear stress imparted on the particles, and then we can really collect very high particles of high integrity, so that we can then work further with those formulations. And this also reflects then in the luciferase expression, for example, an in vitro assay, where we see sort of robustly higher performance of these particles compared to the comparable methods. Now, this process also can give you very highly concentrated formulations. If you work with a high loading and step gradient, you can highly concentrate your formulations and then sort of dilute them down into your formulation buffer to your desired concentration, which is a good process, we believe, for large-scale manufacturing. And this is enabled also by a large dynamic binding capacity, which for the 6 micron OH column is 3.7 milligrams of encapsulated RNA per milliliter of column volume. This then enables you a highly scalable process from a 0.1 milliliter analytical column for like microgram scale up to 40 liter column on the 100 gram scale of purifying your LNPs. To also mention the context of targeted conjugated formulations with for purification, you are also then able to, for example, use this OH column again in the same manner to purify your targeted formulations. And with here, you are then able to either remove your free proteins, which are not sort of connected to the particle, but you're also able to remove free lipid conjugates. And with this, I mean, so a protein attached to the pegylated lipid that is then in a micellar form, which is a common impurity if you use a post-insertion manufacturing mechanism. And in this case, you would be able, the free protein does not bind at all. If it's a lipid conjugate, it does bind to the column, but you can separate it in the gradient, and you can have then your pure purified manually targeted LNP nanoparticles and collect those specifically. So to wrap up briefly, I have introduced the analytical methods and then the challenges we currently face in this field, and hopefully we have contributed a little bit to this with some of the methods developed by our team, and the purification method then also that is able to be used on different scales. So I would like to thank everyone on our team in Sartorius Bioseparations, then Professor Dan Peer for his guidance and collaborations. And now, thank you again. Thank you to Rock. Thank you to the audience. And now we move on to the questions and answer part.
Perfect. Thank you very much, Rock and Tristan, for those excellent presentations. Before we start the Q&A, we're just going to have a quick polling question for the audience, which should be appearing on your screen now. So I'll just give you a few seconds to answer that. Brilliant. Thank you very much for your input. Okay, so I'm gonna invite you both to join me on camera now for the Q&A. Wonderful. See you there. So to the audience, do keep the questions coming in. We're only obviously going to have time for a few today, but we will answer any other questions by email. So if you do have a question, send it in and you'll get an answer one way or the other. Okay, brilliant. So first question I've got here is for Tristan. Can you determine the percentage of the LNPs that are successfully conjugated?
Yeah, thank you for the question. So as I have shown, yeah, you can utilize one of the methods we have developed to sort of monitor that process where you can separate between conjugated and non-conjugated particles. But yeah, do also expect in the future of us to have a two-dimensional system product there where this can be done really robustly in a quantifiable manner.
Brilliant, thank you. Rock, one for you here. What are the limitations to getting high yield in IVT?
So we have shown and actually been produced now around the world, 25 gram per liter is achievable. What has held us back in the past was actually, to my view, lack of understanding of a simple chemical concept that when you synthesize something, you need enough building blocks. So actually, the main thing to crack was to establish the relationship between how much NTPs needs to go in for getting this yield. Actually, the rule is relatively simple. For 25 gram per liter of mRNA, we had to put in 25 millimoles of NTP. It just so works out. There are certain other details about how much magnesium put in and polymerase and so forth, which is all described now, but it's actually simpler than we thought. It was the bottom line. And if we thought it like chemists beforehand, we would have gotten there faster.
Well, it's not often you get to say that something was simpler than you thought, so that's good news. Another question for you, Rock. Does alkali zeta work for any bacterial strain?
We've mostly been working with E. coli, though I don't see a reason why it shouldn't work for other bacterial strains as well. But we see that in plasmid DNA workflows, we're still bound to E. coli, despite certain efforts to move away from it. So all the data that we have is now on E. coli, but perhaps somebody would like to try it on something else, and we look forward to seeing the data.
Well, get in touch if you want to do that. Tristan, one for you. What's the advantage of all these new methods for LNP analytics and purification? Is it really worth implementing?
Yeah, good. Thank you for that question. It's obviously very important. It's always a challenge in implementing something new, especially in implementing something new when you go to a large-scale GMP manufacturing process. But because we are here in a sort of developing field, there is a lot of need for methods that can truly show you what you are missing. So in the first case, if you don't have any of those other orthogonal methods, this type of method can give you a comprehensive tool set in just one step in a relatively fast manner and an automatable manner when you can run multiple of your reactions on this and optimize your process. So in that case, this is very interesting for you. And then in the other case, you will also need some other data later on, and you might start earlier than you would otherwise. Thanks.
Brilliant, thank you. Question here, does it matter if you have a low percentage of supercoiled plasmid DNA going into plasmid linearization for use in IVT?
I think that one's for me. Literature suggests that the answer is yes, and it makes sense. So there's a molecular therapy nucleic acid paper about a year old that suggests that if DNA was nicked in the coding region, then the polymerase will only go up to the nick, and so you will start getting shorter mRNA than you otherwise would. I think we're due to see more and more studies like this, but this is intoning the field towards saying, yes, it is quite important to have highly pure DNA, although it's slightly statistical because not the entire plasmid is coding for mRNA. You've got two strands, so the chance of the nick happening in the coding strand is not 100%. So, but if it does happen in the right place, then you will be making RNA that is not the right RNA.
Yeah, it's an interesting question. Brilliant. Brilliant. Well, thank you very much, Rock and Tristan, for answering those questions. Unfortunately, that is all we've got time for today. So any questions we didn't get to, we'll get back to you by email. This webinar will be available on demand tomorrow, so please do look out for an email from us with the link. We'd also like to invite you to take a short survey to provide us with feedback on today's webinar. The link to that can be found in the resources on the right-hand side of the screen, and it'll also be in the on-demand email. You'll also find an attendance certificate for today's webinar on the right-hand side of the screen too. So all that's left then is to thank Rock and Tristan once more for a great webinar, and thank you to the audience for watching. We do hope you'll join us again soon.